Mr. Chairman, Professor Kumar Srivastava, my colleagues, fellow speakers on the dais, distinguished participants of this conference, I stand here before you to first of all bring you greetings from Nigeria. Like the chairman have said, my topic of presentation is actually impact of microorganisms on sustainable sugar production and of course the sugar industry itself. Microorganisms actually play a very important role in sugar production and the sugar industry. So sugarcane currently actually the economic basis of many countries and planted worldwide. It is the most promising crops for generating clean energy and of course predicted to become the second largest source of energy in the world by the year 2030 and beyond, of course. The global production of this crop is on the increase, mostly because of the rising consumption of sugar and ethanol. Both crop area plantations and yield of sugarcane are occurring to meet the growing demand. Nigeria is one of the sugar producing nations, and you can see from this map, areas in Nigeria painted green are those areas where sugar production is done in commercial quantities. We have a lot of sugar, like I'm going to share some slides with you, but unfortunately what we lack is processing technologies like India, China, and others. I think I'm the happiest person coming to this event in Basandada Institute because I met a lot of indigenous companies that are into the business of processing sugar, making it possible for the value chain to happen. Unfortunately, I must share with you that in Nigeria, despite the massive production of this sugarcane, we only produce it to chew, smoke, and throw away. The concept of value chain is not really being exercised because of the lack of technology and other things. So these are eco-agronomic zones of Nigeria. Every part of Nigeria, actually, one can grow sugarcane. We are so blessed that from one extreme end of Nigeria to another, anything you plant on the soil will definitely grow. This is another map. You can see our position in Africa. We are one of the largest countries in Africa, both in terms of human population and the economy. I will share some pictures with you. This is one of the sugarcane plantations in Nigeria. When I was a small boy at secondary school level, the kind of sugarcane we used to take is different from the kind of sugarcane they are producing now. This one with the pinkish bark and so on. We used to take the one with the greenish or yellowish bark, which is sweeter and better. But today, we have this variety all over that may not necessarily be as sweet. Of course, it has a very big stock, and the biomass production is far better, but unfortunately, like I said, we don't have processing technologies or companies that will go into milling the sugarcane, getting the sugar, and then, of course, ethanol. We heavily depend on importing sugar, and the large chunk of our foreign exchange actually goes on sugar imports. That is sad. That is unfortunate. This is the other variety, that is available in Nigeria, and so on and so forth. Like we have seen from our colleagues that come from China, and like we have seen in some parts of India, mechanization is key. They use a lot of mechanized farming, but unfortunately, again in Nigeria, we still rely on traditional method of farming, where labor is used, particularly women, and so on and so forth. So this actually denies us from maximizing the value of sugarcane. We hope things will change. By the time investors from India, China, and other parts of the country get to Nigeria and invest, so that we can have that value change, we can have an opportunity to have other products derivable from sugarcane as a raw material. It is unfortunate to also share with you and say that even the ethanol that is used in laboratories, that is used in Nigeria and other parts of the economy, is actually imported from India. We don't produce as much ethanol as possible. So I'm calling on the investors to look at Nigeria as a virgin country where you can come and invest so that you make some of these commodities available. Like I told you, the best we can do with sugarcane is to use a sharp knife, peel it, chew it, smoke it, and throw away. Like I said, that is very unfortunate. This is a picture is showing you how we peel and then we chew, smoke, and then we throw away. You can see this picture also showing you what I have just said. That's the best we can do with sugarcane, and it's very, very unfortunate. Nigeria is willing for anybody that will come, either as a company or an investor, so that we can also benefit from other things like that. You can see the farmers, how we load sugarcane to various markets. But it's just for chewing. How I wish that this sugarcane that is produced will be processed. How I wish that we can get ethanol from it. How I wish we can get biofuels from it. So I want investors, like I'm saying, to look at Nigeria as their destination for investment. You can see all this is only taken to local markets. They sell, they buy, they chew, they smoke, and they throw away. The Nigerian government actually have a sugarcane master plan because they realize that sugarcane is a raw material along with cassava for bioethanol production. Our one-time president, President Olusegun Obasanjo, when he visited Brazil, he came back to Nigeria and said we must have a master plan for sugarcane. And that is just on paper. This is the list of some companies in Nigeria. You can see we have only 7 companies that are trying to get the best out of sugarcane as much as they can. The leader among them is the Dangote Group. Dangote is the richest in Africa. He has a lot of investments, a biorefinery, and of course a chemical refinery for processing. And you can see the amount of metric tons of sugarcane they produce per hectare of land and so on. So you can see a country like Nigeria, we need more investments coming from outside rather than relying on, our indigenous. Now, coming to the impact of microorganisms on both crop production and processing, it's quite critical to achieving sustainability, just like my colleague from Sri Lanka have said. Bacteria such as Leuconostoc species and some acid-producing bacilli are usually found in the interior of the cane stalks immediately after cutting. These areas provide sugars, exudates for bacteria to colonize and begin to infect. Other bacteria such as Xanthomonas species and Aerobacter produces mucoids. For some of us that are in microbiology, we know that there are species of bacteria that even when they grow under laboratory conditions, they produce very mucoid situation. And this, to a large extent, is the mechanism they use in causing diseases. Among the fungal species, Phytophthora impestans is notable. And of course, Foliosorium actinomynium, which causes sugarcane smut. Red rot and smut diseases of sugarcane is shown in this picture. Reddish spots on the sugar. Even in Nigeria, where I told you we just chew this sugarcane, normally when you encounter a sugarcane with red spots, actually it did not taste fine. The sweetness is gone, and you have a mixture of tastes due to perhaps sugar acids and so on. So this red smut is not just limited to the surface, as we shall come to see, but it can be deep-seated. Every inch of the sugar cane can be affected. And this, to a large extent, actually reduce the quality of sucrose because the sucrose content is usually affected and so on. So formation of soluble polysaccharides in mildews, also slow crystallization and other problems are attributes of microbes that are associated with sugar processing plants. It is, however, worthy of note that microbes can play a positive role in value addition. What I have just mentioned, these are the negative roles, but if you look at the impact, they can also play a positive role. So in sugar processing industries, for example, Saccharomyces cerevisiae and Zymomonas mobilis are all known to be very active in fermentation of, carbohydrate-based substances derivable from sugarcane into biofuels. They still stand out. These two organisms still stand out in ethanol production. So different strains of Saccharomyces cerevisiae may be available from one region to another. So it is left for us scientists to look and screen them so that we can get the best yeast that can withstand sugar concentration, because ethanol tolerance is one key attribute that one can look for in a candidate yeast that is involved in fermentation. So ethanol is one chemical also with many uses. Many at times we limit the application or the use of ethanol. Here we just talk about the use of ethanol as an energy substance for energy use. But ethanol is very important in cosmetic industries. Ethanol is very important as a disinfectant. Ethanol is very important in pharmaceutical company because it is usually used as an organic extractant to extract active substances from plants and so on. So the value chain, if you use sugarcane processing waste, the value chain is very attractive because we don't only use ethanol for energy, but we can use it for application into pharmaceutical industries and so on. And of course, in the brewing industries, ethanol is the major ingredient of wine and beer. So ethanol as a substance, can also be used as food, because beer and wine, by all classification, they belong to classification of foods that is called beverages. So they are beverages actually. So now, looking at both negative and positive impact microorganisms play in the sugar industry, it is noteworthy that, an evolutionary technology or tool which emanates from bacterial system again, it can be used to solve this problem of negative, or it can be used to enhance the positive problem. And this tool or technology sometimes, and in some bacteria, certain enzymes, nucleases. The technology, the tool I'm talking about is the genome editing. You could recall, apart from the traditional breeding, technique that we use, that actually is more laborious and it takes a lot of time, and the selection of whatever varieties that will come takes a lot of time, genetic modification technology was introduced. Okay, you can get a gene from anywhere, and then you introduce it into any biological system to work. But the problem, this leads to the generation of genetically modified organisms or genetically modified foods. And these genetically modified foods, we all know the kind of controversies they create all over the world. There is problem of acceptance. There is problem of cultural and religious injunctions that is not possible. Some people don't look at the science, but they look at possibility, for example, of getting a gene from a pork or any porcine, for that matter, and introduce it into sugarcane or any food. So there could be religious injunctions, especially from Muslims. Muslims don't eat pork, they don't eat porcines. So that really creates a lot of problem. There are cultural settings that are known to take certain foods only, and by the time you introduce any new thing in the name of GMOs, they really reject it. So these controversies is what led to, the use of genome editing, where restriction enzymes, are used. Genome editing is to look at the gene sequences of the sugarcane or whatever organism you want to edit, and then you now rewrite that gene, or you now trace the genetic sequence that favors infestation from the pest or from the pathogen, so that you can now disrupt that host-parasite relationship. And the moment you're able to, disturb the host-parasite relationship, then the pathogen will not be able to initiate any infection, and so on and so forth. I think this is the latest technology. Here you are not bringing any gene from outside, through transgenesis, but you are editing, the existing gene sequence in the same organism. Happily enough, I will tell you that African Union Development Agency, which has its headquarters in South Africa, secured a project, a multi-billion-dollar project for genome editing technology in order to fight hunger in Africa. And I happily inform you that I'm a member of that team, and we are targeting more than 20 African countries who has accepted genome editing. And we are on. The project will last up to 2026, and we are going to use this kind of technologies to actually solve problem of disease in our food crops, problems of productivity, and so on. So the tool is called genome editing, like I said, or genome rewriting. Here the pathogens of sugarcane and the sugarcane itself can all be edited in order to create. So genome editing is site-specific. Site-specific. That is, only target sequence of the DNA are affected, and modifications are incorporated into genomic DNA using DNA repair mechanisms. For some of us that are microbiologists, we know that naturally some bacterial systems, when their DNA suffer from mutation, they have an inherent ability or capacity to actually repair that damage. So man is taking advantage of that to now look at those enzymes that are involved in DNA repair to now use it in a positive way. We can use the same repair to get defective gene in a crop or in an animal, in order to make good things happen. So there are so many, genome editing methods, but CRISPR-Cas9, as some of you may know, is the most commonly used because it is cheap, it is fast, and it is affordable. Genome editing could play actually a critical role of producing sugarcane varieties or cultivars that could develop resistance to pathogens as well as passing such traits through generations. This is faster, better, and as compared to traditional breeding programs that may take years. So most gene sequences responsible for biomass propagation, because yesterday the keynote speaker on the first day was advocating the use of biomass energy. So for biomass energy, you need substantial amount of biomass. So it depends on what you want as a sugar person. If I want to produce ethanol from sugarcane, then I will actually favor the genes responsible for production of biomass. And if I want to use sugarcane as food, maybe sucrose content is what I look for. So everything, right from disease to growth on an organism, they are all genetic, in nature. You can trace all these activities to genetic level. So it is left for us scientists to sit down and look at how we can make this possible. Science is wonderful, science is possible, and so on. So this can lead to value addition and promote value chain in the sugar industry.
Mr. Speaker?
Yes?
May I request to conclude, please?
Okay, I'm concluding by showing the slides. This is the African Union Development Agency, the program, the project I told you we are working on. And this is some of the concept on molecular genetics. And this is the genome editing. Whatever gene you want, you have to construct it. You have your construct, and then you actually clone it. And even cloning, you should know, is done in microorganisms. They provide a platform for cloning and so on. Then you deliver it into either sugarcane or whatever crop you want to. And then, of course, you screen. And that variety can continue to hand over such properties. So conclusion, it is very clear that the sustainable sugar production and survival of sugar industry, modern approach of pathogen control and genetic enhancement of sugarcane cultivars should be an option. I'm saying genetic enhancement. I want you to note, not genetic manipulation. Many of us are saying genetic manipulation. Manipulation, I consider it as a negative word, so we should try to say genetic enhancement. So I actually give insights that there is a need for this gathering to form a team of experts that will look into these modern technologies because we are moving again with time. I want to sincerely thank you for listening and for your attention. Thank you.
So from audience, if you have any question.
Yeah, I think your question is why is sugarcane production less in my country than India, right?
Yes.
That is one. Okay. You see, like I told you, in my country, there are challenges. One of such is commitment on the part of government to popularize sugarcane production. Leaders in government prepare to import because a lot of money is involved. That at the detriment of local production. But I think the new government that came in last 6 months, they try to revive this policy of sugarcane, local production of sugarcane, because sugarcane is very, very expensive now in Nigeria. I mean, many families actually take their meals without sugar because they cannot afford the cost. So the president now relaunched the sugarcane master plan and policy. So the reason is basically government commitment. And now we have a commitment from the current president, President Bola Ahmed Tinubu, to actually revive that. Thank you.
For increasing the per hectare yield recovery, are you adopting varietal and seasonal programs as per in India?
Sorry, I didn't hear you well. Can you?
Hello?
Yeah, go ahead.
Are you planning varietal planning for achieving the maximum yield?
Nigeria was one of those countries that accept GMOs, so improved varieties of sugarcane. That's why I show you in the picture. The one with pink is actually more popular. So Nigeria really accepts, like India, we accept anything science in order to fight hunger and promote living. Thank you.
Okay, thank you, sir.
Thank you.
Good morning.
Yeah.
As compared to India, why is your country's sugarcane yield is low? What is the main reasons?
Sir, can you come again, please? Sorry.
Procedure to differentiate genetic enhancement and genetic manipulation.
You said I should.
Any specific procedure for manipulation of genetic enhancement and genetic manipulation.
Yeah,
How
what
we can
I'm saying,
differentiate?
what I said in my lecture is that when you alter a gene sequence of an organism, some people usually refer that to genetic manipulation. But you know, this alteration of gene sequence, genome editing, GMOs, is demand-driven. It is for a purpose. It is actually for enhancement. So I caution the use of the word manipulation because I said manipulation is a negative word. So it is better to say enhancement, genetic enhancement, rather than, manipulation. So enhancement is more positive than
That's why
manipulation, yes. Thank you.
So, thank you, sir. Just two small queries.
Okay.
Since the session is on microbes, so just I wanted to know that whether in Nigeria the sugar mills and sugar producers organizations are promoting some microbial products and formulations to the farmers for better yield or for better management, post-harvest management?
Yeah, I think there is a program in Nigeria, Microbial Culture Collection Center. It has been established. So all microorganisms that has useful applications in crop production are actually stored and they are used. And there are a lot of Indian companies like Contech Agro. They have presence in Nigeria right now, producing biofertilizers from microorganisms. And I think they are making a lot of impact in Nigeria. So Nigeria has accepted that.
Okay.
Yeah.
Second thing is related to this.
Yeah.
Do you have any regulation, government regulation for the biopesticides as well as for the genetically modified organisms?
Yeah.
So two parts are there. One is the regulation for biopesticides.
Yeah.
Second one is the regulation for genetically modified organisms or crops.
Yeah, that's a very good question. Nigeria actually, through the parliament, through acts of parliament, established a regulatory agency to look into applications of biopesticides and that is National Biosafety Management Agency to look into the safety and security of those microbes that are used in agricultural applications. So we have that in place, sir. Thank you.